Hello, my name is Daniele Podini. I am the department chair at George Washington University in the Department of Forensic Sciences. I'm also a forensic molecular biology, and today I will be talking to you about some of the research that we've been doing in my laboratory. The title of the talk is Microhaplotypes, the Next Generation Forensic DNA Markers. So mixtures, DNA mixtures, they represent probably one of the greatest challenges in forensic DNA analysis. There's many reasons for that. There's sample degradation, can affect the quality of the mixture, allele sharing between contributors, allele dropout, and PCR artifacts such as stutter. And this is obviously with the conventional DNA marker used in our field, which are short tandem repeats. Massively parallel sequencing of STRs, or short tandem repeats, can increase the ability of deconvolute mixtures. The fact that we can sequence STR and alleles can enable the individualization of some alleles that have the same size but different sequences. It can also allow to minimize fragment length, so we can increase the sensitivity of the assay when the DNA is degraded. And in some cases, it can distinguish between a stutter, so an artifact of a major contributor in a mixture, with an allele that overlaps, normally would overlap with stutter of the major contributor of the minor contributor. And so if we can distinguish those by sequence, that obviously is an advantage in determining, for example, the number of contributors and determining the ratio between the major and the minor. And obviously, in the past couple of years, we've seen probabilistic genotyping kind of really help mixture deconvolution via STRs. But when we talk about massively parallel sequencing, this theoretical advantage that it would offer is actually not so straightforward. Here we have an example of allele 22 of a locus of one of the codis loci D12. And the sequence of allele 22 has, in this particular case, 13 AGAT, and then 8 AGAC, and 1 AGAT. And if you look in position of allele 21 and also of allele 20, there's two stutter. So a minus 4, excuse me, a minus 4 stutter that is actually composed of two different sequences. In fact, during the creation of the stutter, so the slippage of the polymerase, we have a loss. 16% of that stutter is a loss of one repeat. So it goes from 13 AGAT to 12. But the AGAC remain 8, whereas 6% of the stutter, or 6% of the whole percentage, is a slippage at the AGAC. So from one single allele, allele 22, which is the original allele of the individual, the actual DNA contributor, we have two different stutters. And then we have the stutter of the stutter. And then we have the +4 stutter. So with a single allele, we're actually generating another 5 artifacts. So if you can imagine that if there's a minor contributor there, whose DNA is at the level of the stutter, it gets really difficult to interpret which allele is an actual artifact versus one being a stutter, obviously, if it's in stutter position. So although massively parallel sequence has the potential to increase the capability of deconvoluting mixtures, it still has a lot of challenges. And to model these different types of artifacts in probabilistic genotyping, it's very complex. It's not something that's really straightforward. And this is where microhaplotypes come in. Microhaplotypes are a novel forensic DNA marker that was initially thought by Professor Ken Kidd from Yale University. And what they are a set of SNPs within a fragment that can be amplified. So approximately 300 nucleotides or less. And the smaller these fragments, the greater their sensitivity in degraded DNA. And in this fragment that can be amplified, we have multiple SNPs, and the pattern of these SNPs, so the association between the SNP alleles, creates different haplotypes. So normally with the Sanger sequencing, which is the kind of the old sequencing method that's been used for several years, the entire human genome project was based with Sanger, was based on Sanger sequencing. You see right there, you see a CG, a CG, and then two AGs. And we don't know, based on Sanger sequencing, if that first C is associated to the second C or whether it's associated to the G, meaning on which chromosome they are. But with massively parallel sequencing, we can actually separate the two chromosomes because we clonally sequence each individual chromosome. And so in this case, we're able to see that one haplotype, so for example, the one that's inherited by the father, we have a CCAA, and whereas the other chromosome is GG. And so those two are essentially, those two haplotypes are essentially two alleles, but you can have multiple combinations. Okay, so that's what a microhaplotype is. And the advantages of using microhaplotypes are several. First of all, there's no stutter because there's not a sequence that's repeated multiple times. As the polymerase amplifies that region, there's no slippage, and obviously they can be selected appropriately. So the efficiency of the amplification is high. They are SNPs, so they're based on SNPs. So the mutation rate is a lot lower than short tandem repeats, and so they can be very useful in relationship testing. They can be useful because there's no stutter, and because there's this because there's no stutter in an imbalanced mixture. You would not have the issue that you have with STRs, where stutters can overlap with alleles of the minor contributor. That issue is absent. Modeling in probabilistic genotyping is much easier because you don't have the stutter issue at all. And also, all the alleles within the same locus have the same size. With STRs, you may have alleles that are much larger than others because they may have a lot more repeats. And so this gives the larger allele an amplification disadvantage if there's inhibition or if the DNA is degraded. So you don't have that issue. So there are several advantages. There are some disadvantages, of course. For example, they are less polymorphic than STRs. So you need to increase the number of loci that you amplify, that you co-amplify in multiplex to get the same power of discrimination that you have with STRs. So designing a multiplex assay may be more complex. And obviously, although I don't have it on this slide, they're based on massively parallel sequencing, which is more expensive, at least right now. Going forward, I think massively parallel sequencing will improve, throughput will increase, there will be a better way to increase the number of samples that you can analyze at the same time. And so it's very promising. So as I said earlier, Professor Kidd is the one that thought of this, and he started publishing paper. The first paper was, as I show here, in 2013, and he was the one that mostly published for the first 5 years on this. But then there was an explosion of papers, and there's actually more that have come out and are coming out, including a couple that we submitted, and we're waiting for the reviewers to get back to us. So the community has really, there's consensus in the community that this is actually a good forensic DNA marker. So we received funding from NIJ to develop an assay based on a panel that was identified by Dr. Kidd in collaboration with others, including our group. We selected a subset of this panel based on two parameters. One is AE, or effective number of alleles, and the other parameter was is informativeness. So the effective number of allele is essentially how it tells us how discriminating is this particular locus. So essentially the distribution of the alleles that this particular locus has, and how balanced is the allele frequency of the alleles that are identified for this locus. The informativeness, instead, tells us how unique are certain alleles to certain populations. So, in essence, AE allows us to discriminate individuals within the same population or across population. Whereas the informativeness, a higher informativeness, gives us the ability to discriminate between individuals from different populations. So we selected a panel based on that, and we analyzed multiple samples. The throughput, the process of the analysis was first the library prep preparation. We did both manual and using students as part of their projects, and also automated library prep. Then we did what's called chip templating. Well, the templating and loading on the chip. So the libraries go through a process of emotion PCR, and then eventually they get purified and loaded onto the chip. And then they were sequenced on the Ion S5. So this is the Ion S5 MPS platform. Then we worked on pipelines, for bioinformatic pipelines to process the data using some that were provided by Thermo Fisher's, others that we developed. And the final output, in this particular case, kind of mimics what the conventional STR analysis looks like. So we have in this case, we have a bar as opposed to having a peak, and the height of the bar represent how many individual reads of that particular sequence have been found. And at the bottom, you see A, G, and GG are the two alleles, meaning A and on one chromosome we have at the first SNP an A, at the second SNP a G. In the other chromosome we have two Gs. And these are not necessarily contiguous. They can be separated, as I showed you in the previous slide. And we did several studies. So I'm going to go through some of them here. I don't have time to go through all of them, but if anybody's interested, they can contact me and we can talk about it. So we did a mixture study. Oh, the first is the sensitivity. So the sensitivity that we were able to obtain, we obtained full profiles down to 50 picograms of input DNA, which is very similar, if not better than most of the conventional STR kits that are commercially used by prime labs right now. So we developed multiple mixtures, starting from 10 to 1 to 20 to 1, 40 to 1, even down to 80 to 1. And then we did multiple contributors. And this slide kind of shows you the distribution of types of mixtures that we looked at. We obtained full profiles for most of them. Obviously, as we increase the ratio between the major and the minor, we're inputting always less amount of DNA of the minor, given that the overall amount of DNA, as you can see on the right of the slide, is 1 nanogram. So as you increase the ratio between the two, you're always putting in less and less of the minor contributor. So we started seeing some, as expected, some allele dropout in the 20 to 1, a lot of allele dropout in the 40 to 1, and the complete dropout of the minor contributor in the 80 to 1. Which was expected. We analyzed those mixtures in multiple ways. So we used our microhaplotype assay. We also used the conventional global filer kit with the capillary electrophoresis, the 24 loci assay that is been validated in many laboratories. And then we used the new Precision ID Global Filer NGS STR panel version 2. And that includes all the 24 loci that are in the conventional CE Global Filer, plus an additional set of loci that increase the power of discrimination of the assay. So we analyzed the same mixture with the same amount of DNA in these 3 different ways. And here are the data. So this is an example. We're talking about sensitivity, a 20 to 1 ratio. As we expect, we see some dropout of the minor contributor with conventional CE. So for example, at locus D3, we see the 16 and the 18 of the minor contributor that are very low. WA, we see the 18. Then we see some dropout, and some are masked. So for example, at locus CSF 1PO, the minor contributor is homozygous 12, which is obviously masked, in this case by the major, who is 912. The 40-to-1 ratio, as we expect, we don't see any of the minor contributor with conventional CE. When looking at the same samples but tested with massively parallel sequencing global filer kit, we end up getting a little bit more information of the minor contributor. Again, just to navigate you through this plot, the height of the bar represent the individual sequences, and the number underneath each bar represents the allele call. And these are sequenced, meaning the software reads the sequence that's generated by the sequencing on the chip, and then it counts the individual repeats, and then it develops this output. So it's kind of an really artificial output, as opposed to the peaks that we were seeing in the previous slide, which are, the height of the peak is actually a consequence of the amount of fluorescence that is generated by the PCR product as it goes through the capillary. Almost complete dropout. So at the 20-to-1 ratio, we see a lot of the minor contributor, more than we were seeing with CE. In the 40-to-1 ratio, essentially we see complete dropout. We see a few elevated stutters, like in the D4S 2408. We see a little bit of the 10 that is slightly above the stutter threshold, but this cannot be considered reliable because this is a new kit. We hadn't done a thorough actual internal validation. So we don't have stutter threshold. We're just using the ones that are that were given by default. And again, there's not been enough processing of this kit to be able to determine whether that 10 is actually above the stutter threshold, meaning there's the minor contributor that's increasing the read count. Okay. Now, the same mixtures with microhaplotypes, we see a significant amount of alleles of the minor contributor at the 20-to-1 ratio, where at the 40-to-1 ratio, obviously we have more dropout, but we're still detecting a good amount of alleles of the minor. 54 to be specific, and 22 alleles of the minor contributor. And we'll see later, I have a slide showing that the random match probability that is generated with just these 22 alleles of the minor contributor is sufficient, is strong, is pretty powerful. We're getting, 1 in a million or around that area. So we're going from a profile that's analyzed with CE that yields nothing, 40-to-1, we didn't get anything, to something that can give us a random match probability of 1 in a million. So it's a very powerful technique. Now, the idea here, okay, we've seen that it actually has the potential of, I'm not going to say replace STRs, because STRs are here to stay. There's an enormous inertia of, the convicted offender database that has, millions and millions of profiles, STR profiles. There's forensic profiles within CODIS that are obviously potentially useful to identify offenders that haven't been officially typed yet. So STRs are here to stay, but it has the potential to enhance mixed deconvolution or to complement STRs. But in order for this to happen, we have to make them attractive to forensic scientists. So how can we make them attractive? So first of all, there has to be user-friendly software that makes it easier to look at these samples, and it has to be robust. Developing a bioinformatic pipeline is not as easy as I thought, at least when I got into this field. Converting so the conventional output is pretty straightforward from CE. So the amount of fluorescent generates a signal that's detected and kind of plotted into that graphical representation that we're so used to. The bioinformatics, there's a lot of noise. There's so many more samples. It's something that cannot be done manually. So it's a lot more complex. Then we need allele frequency, because if we allele frequencies are what allows us to determine the frequency of a profile. So we have a match between the evidence and a suspect, but we can't just say it's a match. We have to provide a statistical significance of this match. Or call it random match probability, we could call it likelihood ratio, it depends. And then they have to be amenable to probabilistic genotyping. The software is complex enough. The number of loci are much greater than with STRs, because you want to obtain, as I said earlier, you need more loci to obtain the same discrimination power. So you can't really do it manually. And as we've seen in the community, everybody's shifting to probabilistic genotyping because it makes things so much more consistent, much more straightforward. There are challenges to it, but this is something that would be a must for microhaplotypes to be implemented. And finally, there has to be an international consensus on the final panel that we want to use as a community. And that is really crucial. And I'm not going to talk about it now because I really haven't been working on this right now directly, but it's something that's extremely important, because that is what's going to allow the companies, the manufacturers of these kits, these molecular biology kits, to then decide whether it's worth investing their resources in developing a panel. So there has to be consensus between the community on which are the loci to type. So user-friendly software. So here is an example of a kind of an alpha version of a plugin for Armed Expert, and this is thanks to Brian Young. You see his picture at the bottom on the left, and obviously this doesn't tell you much. I zoomed into one of them. So here is just a plotting of the results of a set of microhaps of our panel. Again, the height of the bar represents the coverage, meaning how many sequences have been identified with that particular pattern. The label on the top shows you the allele call, and right now they've been labeled with a particular code, but that can be adapted to anything we want. The locus name is at the top, and the two alleles are what you see at the bottom. So the bar represents the individual allele. So as you can see now, there's some imbalance. This is a single source sample, so ideally you would want one peak when the individual is homozygote, or two balanced bars, I should say, when the alleles when the individual is heterozygote. But we see, like in the center, where it says unique identification code, right in the center, you see that there's two alleles for that locus, and they're significantly imbalanced. There's almost twice the amount of one versus the other. So that's a bioinformatic issue. So we have to optimize the pipeline in order to clean better and to identify which ones are the actual alleles. And you see a couple of those also. Also, we want to try and that's more kind of a PCR primer concentration issue. Try to balance all the different loci, so that the coverage is similar across the loci. But this is an example of how we would want a software. So you kind of select the sample, you run the software, and you get these bars. And then obviously the output is that they have to be exported into a table that can be used for comparison. And if it's a mixture, there could be some initial manual interpretation, but this is a good way to visualize the sample. And it's attractive to old school DNA analysts that are used to looking at peaks, right? The bioinformatician doesn't really look at this anymore. So this is really for us molecular biologists or DNA analysts. In terms of allele frequencies, we've generated, we've sequenced several samples for the purpose of generating allele frequencies for the major U.S. populations, so African American, European Americans, East Asian Americans, Southwest Hispanics, and we've typed also over 800 samples, 14 different populations throughout the world. So we have the allele frequencies that can be used already on the set that on the 74 haplotypes that we've analyzed. And we've also tested how well we could use microhaps to predict the ancestry of an individual. So for that, we used the allele frequencies that were generated for many more populations across the world by Dr. Ken Kidd and his lab. And here's an example of how ancestry prediction could be done. So here, these are individuals of African descent, and what you see is, so the plot represents the frequency of each individual profile in all the populations that are listed on the top of that graph. So these are 20 individuals of African-American descent, so that's their biogeographic ancestry. And as you can see, the frequency of their profile is higher in populations from Africa. So as you move from left to right, at a certain point, there's a drop which corresponds to allele frequencies that were from individuals of European descent. And although on the graph there's not much difference, but each one of those lines, those horizontal lines, is 3 orders of magnitude. So in essence, as you drop, what is it, 1, 2, 3, or 4 lines, it's like 12 orders of magnitude difference in terms of the frequency of that profile in that population. So you clearly see a pattern of individuals of African-American descent. These are Europeans, and again, you see that their frequency bumps up when we're using the frequency, the overall frequency of the profile comes up when we're looking at when we're using allele frequencies from European individuals. These are East Asians, and these are Southwest Hispanic individuals. And as Southwest Hispanic are actually an admix between Europeans and Native Americans. And you see that the frequency goes up in both, which is what we would expect. Here's an example of those are kind of the averages of the plots that I showed you before. And you see also, and this is a single individual in purple that was a profile from a Native American individual. And you see that the frequency is relatively low across Africa and Europe. It goes up a bit in East Asia, and then it goes up again, significantly up, obviously, when we use allele frequencies from individuals of Native American descent. So this shows you that they actually have the potential to provide some ancestry information. And here's kind of a different approach. So we're using a multivariate statistical approach that uses a logistic regression and PCA combined. And these are some of our samples. And as you can see, the plots, the individuals are well separated into different parts of the plot. So this is another approach. So you would add an unknown individual, the software would plot it in one of these positions. And so you could use this as a method to predict the ancestry. Now we're still working on this part, but it definitely has potential. And this is in collaboration with the Carabinieri group of Rome and the University La Sapienza, who've been using this statistical approach on STRs, showing some really promising data. And so because microhaps carry more ancestry information than STRs, it is like, at least our preliminary data, we're seeing that it has the potential to really help in this process. This is a kind of an example of combining mixture interpretation and ancestry prediction. So on the left, we have the likelihood ratio of the observing this particular profile if it's an individual of European American descent versus, in blue, an individual of African American descent, meaning it's 10 times to the 16 times more likely to observe this particular profile if it's a European, if the DNA came from a European versus an African American. Whereas the red bar shows us that it's about approximately 1,000 times more likely to observe this profile if it's an individual of European descent than if it's an individual from Hispanic descent. And again, European versus Asian is a lot higher. So, and this is for the alleles of the minor contributor in the 20-to-1. This is to the left. And to the right, it is for the 40-to-1. On the right side, you see the random match probability. So you see that the random match probability for the profile is in the, and that is kind of the average, is in, the 10 to the 19. So we're talking trillions of trillions. So it's a very, very high number. And this is for the minor contributor of the 20-to-1. The minor contributor of the 40-to-1, that profile is around 1 in a million. So it's still pretty strong. Although the ancestry information that we're getting out of the 40-to-1 is not as informative. The conclusion here is that we could say that the minor contributor of the 20-to-1 mixture is most likely of individual of European descent, possibly Hispanic descent, but we can probably exclude or assume extremely unlikely that the individual is of African American descent or East Asian American descent. Probabilistic genotyping, and this is the research that we're currently working on. So taking the data, that's the output data from our bioinformatic pipelines, and then throwing it into probabilistic genotyping to see if it works. And for this, we've been working on using LR-Mix Studio, which is a semi-continuous model that basically doesn't look at the height of the peak, so the amount of DNA, but simply looks at the presence or absence of alleles. And then we, in collaboration with Dr. Charles Brenner, we've been using DNA View mixture solution, that was adapted by Dr. Brenner to importing microhap data. And I don't have many examples here in the interest of time, but we've demonstrated that it actually works pretty well, and microhaps are a good tool that can be easily adapted to probabilistic genotyping because of its characteristics that I mentioned earlier. You don't have to model stutter, fragment. You don't have to model size between alleles of the same locus as you have to do with STRs. And so it's actually working pretty well. And this is an example of a 5-person mixture where the hypothesis are that either it's a mixture between 2 individual B and C plus 3 unknowns, or a mixture of 5 individuals B and C but plus 3 unknowns, or B plus 4 unknowns. So essentially the person of interest is Mr. C in this case, which is, in this particular case, was the intermediate contributor, so the one that's at 5. And so a mixture solution is able to very effectively deconvolute the mixture and attribute the alleles to the different individuals, particularly the intermediate person. And it gives us, 1 times 10 to the 78 likelihood ratio, which I don't know even how to say it in terms of trillions of quadrillions or whatever it is. Whereas LR-Mix Studio gives us 8 times 10 to the 3. So you would say it's 8,000 times more likely to observe this mixture if it's a mixture between B, C, and 3 unknowns than if it's a mixture between B and 4 unknowns. And we expect that LR-Mix Studio doesn't have the same power of deconvolution because it's not using peak heights or, in this case, coverage. So this kind of demonstrates that these are that microhaps are a good marker for probabilistic genotyping. Now we wanted to take it a step further. So we've been focusing on the SNPs that define the microhap. So the ones that initially Dr. Kidd identified. This is an example of an amplicon, and those red bars represent the location of the individual SNPs. So these are the ones that define the locus. So the profile, AAGG, or A at the first red bar, A at the second red bar, G and G at the third and fourth red bars. Okay, but there's a lot of real estate there, a lot of sequences that we've initially been ignoring. So our initial pipeline was actually only focusing on the 4 SNPs. But there's a lot of potential genetic variation that can occur there. And the greater the number of loci that we're analyzing, so the greater the number of this genetic real estate that we're looking at, the greater are the chances that an individual has a rare SNP that is identified. So here is an example. These are the 2 microhaps that define the 2 SNPs that define the microhap. But then if you look at all those potential SNPs that have been identified, there can be variation there. And some of it may be not informative because maybe it's completely linked with the allele that we are already genotyping, but others may not be. And here's an example of an individual that is homozygote GA based on focusing on the 2 SNPs that define the microhap. But when we actually go and sequence the individual, we see that there's another SNP variation right there that is extremely rare. As you can see, it's been identified very rarely in individuals of African descent, but it's been virtually not seen in all the other populations. So this individual now becomes heterozygote. So it goes from homozygote to heterozygote. Not only that, but it increases the power of discrimination of this profile incredibly. So in a mixture, for example, we would be able to, again, discriminate but by adding this rare allele, which would be GGA, we increase the power of discrimination and the chances of also identifying the number of contributors to a mixture. The next frontier of these microhaps is already being kind of addressed by Dr. Kidd, who's now focused on identifying microhaps that are very small. And the advantage of these small microhaps is that they're going to be a lot more effective on extremely degraded DNA. So STRs go from approximately 100 onward. And with massively parallel sequencing, maybe you can stay within, between 100 and 200, 250, something like that. So DNA is not that big fragments, but if the DNA is significantly degraded, you're not going to get a good profile. If you focus on fragments that are smaller than 50 base pairs, you can actually increase the discrimination power. And so here's a structure plot of 108 microhaps that are smaller than 50 base pairs. And as you can see, they have a very good informativeness, and although it's not shown in the slide, they also have a good AE. So they have a great potential for typing degraded DNA where STRs would fail. So here are the various things that we're still working on, improving our mixture deconvolution, ancestry prediction, degraded DNA. We're looking at relationship testing, how effective they are, and not only father-child, but siblings, step-siblings, cousins, and so forth. We're in the process of publishing the allele frequencies and the mixture studies, and also optimizing the bioinformatic pipeline, which is not a small undertaking. With that, I also want to thank all the people that worked the data you've seen. There's the GW students, particularly Fabio Oldoni, who is the one on the right of the picture on the bottom right. You see me, Ken, Kid, and Fabio, who's done a lot of work here. And also all the students of the program. In this picture, there's Lina, there's Chiara, Satya, Aishwarya, Drew, myself, and Fabio again. So they've all helped a lot, and other students that I won't, there's too many to mention, but also and especially NIJ for funding the research that we've been working on. That without their help, the help of Thermo Fisher, Niche Vision, we wouldn't have been able to generate this data for them. Thank you so much.